Hello. What you see here is a group of people that are interested in microtubules, but all from a different angle. And the goal of the seminar was to agree on something that we're all interested about. And people can tell you if we managed.
We managed.
So Filippo,
Well, no, the unique aspect of this meeting was to bring together communities of people that, although they share a common interest in tubuling, they normally address this question with very, very difficult different techniques and at different scales, different model system, and normally they don't talk to each other too much. So we brought them together to be able to hopefully cross-fertilize the ideas that we have from different fields.
So for those that are not molecular biologists, tubulin is a protein, is a component of the cell that has the capacity to self-associate, making these amazing structures called microtubules. They are microscopic in nature, but they are tubes that play many different roles. And we are all interested in how they're made and how they're utilized in a cell to do different things. So we have examples of neurobiologists that are visualizing or learning about how these microtubules serve as freeways along which motors move cargo from one end of the neuron to the other. And others are interested in how tubulin self-assemble into these amazing structures that are called microtubule mitotic spindles that align and segregate chromosomes. So very, very different functions and studied by very different means. I personally like to visualize things, but there are many ways in which we can learn about these polymers.
The living cell is the unit of life. A single cell can do almost everything life can do. It can grow, it can move, it can divide. And all this is possible only because it has a very elaborate machinery inside, which we call the cytoskeleton, the skeleton of the cell. So altogether, we're trying to understand how this works. It's a lot of different techniques to be able to observe and to analyze the biochemistry and to understand how all of these molecules together produce, for instance, movement or division machinery or growth. And all this sustains life in general, all the life we can see on this planet.
As a fundamental part of life, every change in this machinery causes important diseases, and many of them interest the nervous system. And by understanding the molecular mechanism from the single atomic structure to the cellular structure to the organism, we are really trying to address and find a new cure to address this pathology.
Microtubules in cells can generate large assemblies, and sometimes these large assemblies are highly structured and follow peculiar geometries, symmetrical structures. One of these assemblies is the cilium, which is the cell antenna, allows cells to communicate, and also allows many cells to move fluids over tissues. And this is an interesting model to study how microtubules can form moving structures and beating shapes and self-assembly in very organized forms.
I have probably been in this microtubule field longer than anyone else in the meeting. For me, what was so exciting about this seminar was hearing some definitive answers to questions that have been in the field for years and years, but more exciting, a view of the future, of unsolved questions, and amazing new methods that will allow us to address, probably younger people than me, address questions that I thought would be impossible to solve. We saw, the first pictures of certain proteins sticking to microtubules that look nothing like we expected. So I think I'm excited for what's been achieved, but more excited for the future.
Good doubting, Thomas.
Yeah, one of the most interesting aspects of microtubules is how they are organized in whole cells. And I'm, for instance, studying the mitotic spindles, and the fascinating part is how about 20,000 microtubules are organized in a cell to build a spindle and to segregate the chromosomes. And in parallel, colleagues are doing simulations, and we want to understand at the same time how the spindle is working on a biophysical level.
Signaling to the microtubule cytoskeleton.
Mhm.
And even though each cell has in common this particular type of cytoskeleton, which is made out of microtubules, we really don't know what are the signals that organize it and how it's regulated, depending on the specific function of that each cell in our body has.
So to tackle those different questions, we also need to push the boundaries of experimentation and technology, and this is what some of us are trying to push, to really invent new ways to approach those questions, and really we can learn from this fundamental aspect. We can also have implication in disease or have some translational aspect.
The study of microtubules is very old, older than anybody sitting here. But what was exciting to me about the seminar and bringing this group of people together in this beautiful place was that we were able to really summarize what important developments that have answered now, I would say, salient questions that have been in the field for many decades, that were possible by new technologies that were introduced in the last 2 years. And also, I think we had the time and the inspiration in this place to discuss about new concepts and new technologies and new ideas for the future and for the new generation that's going to enter this field.
What we'd like to do is to try to understand how the cytoskeleton is built, and it builds itself. It consists of many components, and what we do is we purify or isolate a small number of them, and then put them together and try to rebuild parts of the cytoskeleton. And by doing this, we try to understand the rules, how the molecules find each other in the right place.
By contrast to Tim, who has been in microtubule research for the longest time, I think I'm definitely the one in microtubule research for the shortest time. And this group has been wonderful, amazing. The kind of things they do at different levels, like some of others were describing how they're studying how these assemblies come together. Some of them are studying how these assemblies function in cellular level, and some of them are looking at the disease relevance and stuff. So for me, it has been fantastic learning experience, and towards the end we've just been like brainstorming crazy to bring together what we think as a group is important for next 5 years, probably 10 years, or maybe we meet again and look back at the same questions. But overall, fantastic meeting, and thank you to Letrai also for giving us the funding and making this meeting possible. Thank you.
But first of all, I think I was very lucky to be here for these days. I've learned a lot of things. But one thing that I can say, as I think I've mentioned actually before, is that this field of research, as it's been around for quite a while, can be considered by some or defined by some as mature, which can be interpreted as when there are not so many novel concepts or ideas by only just trying to figure out the details of some processes. And I think I can say that that's not the case. It's a very lively field, and I think it's actually full of ideas and intellectually challenging. So.